All right, welcome to the second video for Chapter 13, which is about biochemical signaling. And in this video, we're going to focus more on signal transduction. And so, once again, signal transduction is what happens to help transmit signals into cells and ultimately tell the cells to carry out a variety of different functions. So first we'll talk about some of the general principles of signal transduction. Then we'll move on to a couple of examples including receptor tyrosine kinases, or RTKs, G-protein coupled receptors, or GPCRs, and finally we'll move on to some of the signal transduction pathways, with our one example being the AC pathway. So first, let's just talk about some of the general principles of signal transduction. So in the previous video, we talked about how hormones might travel through the bloodstream, and they are basically a signal molecule. Once they arrive at their target cell, they act as a ligand by binding to a receptor on the cell that's specific to that specific hormone. And once the ligand, which is the hormone, binds to that receptor, a conformational change happens in that receptor. And because this is a transmembrane receptor, that means that this information that came from the outside of the cell ultimately will give information that is moving to the inside of the cell. And then once that conformational change happens inside of the cell, then a signaling cascade starts, which involves secondary messengers, which can ultimately affect a number of different intracellular targets to ultimately affect a number of different processes and functions that a cell might carry out. So we've seen this before, so one really important concept in signal transduction is phosphorylation of either serine, threonine, or tyrosine. It's really important, as we mentioned, for enzyme regulation, for turning enzymes on and off. And this can be really important for signal transduction because as each component within that signaling cascade is turned on and off, this often happens through the addition and removal of phosphates. And so just as a review, we mentioned before how kinase enzymes are really important for using ATP to phosphorylate enzymes or whatever their substrate, their target is. And phosphate enzymes are really important for removing phosphate groups from their substrates, which again are often enzymes. And so you can imagine during a signaling cascade, you might have one enzyme which gets turned on by a kinase, it becomes activated because it's phosphorylated, and then once it is activated, it might go on to act as a kinase for the next enzyme that's in that signaling cascade, and so on and so forth. So let's get into some examples of these receptors and the beginning of these signaling cascades. And so our first example is going to be receptor tyrosine kinases, or RTKs. And so receptor tyrosine kinases are, as the name suggests, they start out as receptors, right? So this means that they are going to be transmembrane proteins that have an extracellular domain that's able to bind to a ligand that comes from somewhere outside the cell. Then there's an intracellular domain that's going to help to give information to the inside of the cell to help carry out a particular function. So some of the ligands that can help to activate receptor tyrosine kinases include polypeptide hormones, as well as different types of growth factors. And once the ligand binds to the extracellular domain, which we see here, the two monomeric receptor tyrosine kinases will dimerize. They'll come together to form a dimer. And once they come close together in space, the tyrosine kinase domains, which are on the intracellular part of the RTK, are brought together closely in space, and they're able to phosphorylate each other, right? So this is a receptor tyrosine kinase. Its job is to add phosphate groups to tyrosines, hence the name. And so this process is often called autophosphorylation or trans-autophosphorylation because one tyrosine kinase domain on one of the receptor tyrosine kinases will add phosphate groups onto the other one, and vice versa. And so ultimately, you'll end up with the intracellular domains of these tyrosine kinases getting decorated with all of these different phosphate groups. And depending where these different phosphate groups are added, they might act as targets for different second messengers, right? So different other proteins that are inside the cell might bind to different areas of the receptor tyrosine kinase, which will ultimately help to start a variety of different types of signaling cascades, which depends on the ligand that bound from the outside of the cell. So that is very briefly sort of receptor tyrosine kinases and how they are able to initiate a signaling cascade. Another type of receptor that can help to initiate a signaling cascade is the G protein coupled receptor, or the GPCR. So GPCRs are made of transmembrane alpha helical proteins. So as you can see from this diagram, there are 7 transmembrane domains that are alpha helical. And so there's an extracellular component as well as an intracellular component. This particular one is also lipid linked, which you can see in this picture here. And then the effector for GPCR, or the ligand, right, which comes from outside of the cell, can include a number of different ligands, including peptide and protein hormones, nucleosides, nucleotides, calcium, and biogenic amines, among other things. And then once this GPCR is activated, it is going to go on to activate different components that are located inside of the cell that interact with the intracellular portion of the GPCR. And in particular, what the GPCR will activate on the inside of the cell are G proteins. So this might, be helpful based on the name, right? So a G protein coupled receptor means it's a receptor that is coupled to a G protein. And so the G proteins that interact with GPCRs are heterotrimeric, meaning they have 3 different subunits that are all together. And so the subunits on our heterotrimeric G proteins are alpha, beta, and gamma. And G proteins are called G proteins because they bind to GTP. And so in the case of heterotrimeric G proteins, it's the alpha subunit that binds to GTP, and you can see that in here. So in this diagram, the alpha subunit is shown in pink, and you can see how GTP is bound right in there. You also see the beta subunit a little bit above there, which is shown in the sort of rainbow color, and then there's the gamma subunit, which is shown in that orangey kind of yellow color. And that's kind of a side view. On the right over here, you can see this kind of top-down or bottom-up view, where the beta subunit is still rainbow and the gamma subunit is still yellowy orange, but the alpha subunit is now shown in gray. And so these 3 subunits can each have a particular function that they can carry out. But overall, these heterotrimeric G proteins can be either stimulatory or inhibitory, GS or GI. Meaning that they can activate or inhibit some of the components that are downstream, that are further along in the signaling cascade. So let's get more into how this signaling process actually works. So we have shown in this diagram our GPCR, our G protein coupled receptor, with its 7 transmembrane domains sitting right in the cell membrane. There's a signaling molecule, which is a ligand that comes from outside the cell. And once that ligand binds to the G protein coupled receptor, it's going to go ahead and interact with this G protein, which initially is inactive. And when it's inactive, the alpha subunit is bound to GDP, and that is what makes this G protein inactive. Once the ligand binds to the G protein coupled receptor, it's able to interact more closely with the heterotrimeric G protein, and the GDP gets exchanged out for a GTP. And once GTP is bound in the alpha subunit, it is activated, and the alpha subunit will separate from the beta and gamma subunits, which are shown in the orangey color and the green. And these proteins are tethered, as you can see, in the cell membrane by lipid tethers, which we discussed a little bit in previous chapters. And so the alpha subunit separates out from the beta and gamma subunits, and they can each individually carry out different downstream functions. And they can start up different signaling cascades that ultimately result in a variety of cellular responses. And what we're going to go into a little bit more detail right now is the adenylate cyclase, or AC pathway. And adenylate cyclase, which is also called adenylyl cyclase, is shown here. This is another transmembrane protein. That's an enzyme. This is sort of summarizing what we just saw. So in this case, we have our ligand, and in this case it's adrenaline, is binding to the adrenergic receptor, which is one type of GPCR. And when it's activated, that GDP that's bound to the alpha subunit in that heterotrimeric G protein gets swapped out for a GTP, and then that alpha subunit is able to separate from beta and gamma and head over to the adenylyl cyclase, which is, as I mentioned already, also located in the cell membrane. So this lipid tether that's on the alpha subunit helps it stay in the cell membrane, so it's able to go find the AC once it gets activated. And then once the alpha interacts with the AC enzyme, it is able to get activated and perform its function, which is converting ATP into cyclic AMP. And we show that in a little bit more detail up in this upper right-hand corner. So on the left side of this reaction, we have ATP, which you should be familiar with by now, which has 3 phosphate groups attached to this ribose. And then adenylyl cyclase or adenylate cyclase, same molecule, same enzyme, 2 names, cleaves off a pyrophosphate and helps that one remaining phosphate form a bond with carbon number 3 on the ribose, helping to form cyclic AMP, which you might have also seen written as CAMP. And ultimately, cyclic AMP is the second messenger, which goes on to activate the next step in the signaling cascade, which we're showing here is turning on another enzyme, which is called pKa. So pKa, when there's cyclic AMP around, gets activated, and pKa is a protein kinase. It's protein kinase A. And then it goes on to perform its function, which it's a kinase, which, as you know, is going to phosphorylate its targets. And ultimately, this is going to can activate a number of other further down the signaling cascade. Ultimately can activate or inhibit a variety of enzymes, as well as transcription factors, which can give instructions in the nucleus to help transcribe and ultimately translate a variety of different proteins that can help the cell perform a variety of functions. Whoops. So apologies. So now that we've gone over a few different examples of sort of how signaling cascades can get initiated through RTKs and GPCRs, let's kind of bring together what we learned from the first video and what we learned in this video. So we have a number of different hormones that can act on different cells and help the cells perform a variety of functions. So let's start with our pancreas up on the top here. So the pancreas, when energy levels are low, when blood sugar levels are low, it's going to secrete glucagon. Glucagon can travel through the bloodstream, and it's going to interact with receptors on cells, and specifically it will only interact with glucagon receptors. And so once glucagon binds to its specific receptor, which happens to be a GPCR, right, that 7' transmembrane receptor that interacts with the G protein, which activates, then the alpha subunit gets a GTP bound to it, which helps it find the adenylyl cyclase, which ultimately helps to make cyclic AMP. And as we talked about, cyclic AMP will activate pKa. And then in this case, pKa we're showing is going to help activate the enzyme that helps with degrading glycogen. And as glycogen is broken down, the cell is able to release glucose monomers, which ultimately are secreted or sent out of the cell through the GLUT2 glucose transporter. And then glucose can then be sent into the bloodstream to be used by whatever tissues are in need of glucose. On the sort of similar but different, we have adrenal glands. And we talked about how the adrenal glands secrete epinephrine. Epinephrine interacts with another G protein coupled receptor, beta adrenergic receptor. And that's going to have a very similar effect to the glucagon receptor, but for a different reason. So we talked about how glucagon is secreted in response to low blood glucose levels when energy is needed. Epinephrine is secreted sort of in expectation of the need of more blood glucose during the fight or flight response. And so a lot of energy might be needed to help with that stress response. And so epinephrine is sort of helping to ultimately activate that same pathway to release glucose, but for a different reason. And and then we have our other hormone, insulin, which is also secreted by the pancreas. And insulin acts on its own specific insulin receptors, both in the liver and in muscle. And so insulin, as we talked about, is released in sort of the opposite circumstance. And the insulin receptor is actually a receptor tyrosine kinase. But ultimately, it's going to do the opposite it's going to tell the cell to sort of perform the opposite function as glucagon did. It's going to activate this receptor tyrosine kinase that's ultimately going to help promote glycogen synthesis to help store energy. And so it's also going to encourage glucose to enter the cell so that it can be made into glycogen. And it's going to act both at the liver as well as at the muscle for the same way. It's going to help glucose enter those muscle cells and help it to build up glycogen. And so we can see that these different types of hormones are acting on both liver cells and at muscle cells, sort of when either in a low energy or a high energy environment, or particularly in the case of blood glucose, right? And so hopefully that sort of gives you a bigger picture view of how hormones are functioning to send signals from one part of the body to the other to help ultimately different tissues and organs perform a variety of functions. So now that you've watched this video, you should be ready to answer the Chapter 13 signal transduction practice questions.